Hello class, my name is Chris Grippo,
And I'm Adam Ranellone.
and together we made a small presentation about the unique properties of calliotoxin and how it functions in a mammalian body.
So this is a general overview of the topics that will be covered in this presentation. First, we'll take a look at the long-glanded blue coral snake. Then we'll explore how its venom affects our bodies, particularly our ion channels, and lastly, how it was isolated and then characterized.
The blue coral snake, or Calliophis bivageris, is part of the family Elapidae and is native to Southeast Asia, particularly in areas of Indonesia, Malaysia, and Singapore. Its distinguishing marks include a bright red head, tail, and belly with a white stripe on either side of its dark body. One of the main reasons its venom is so potent is that its diet consists of other venomous snakes. As such, a strong venom would ensure quicker incapacitation of the prey before it could bite back. A unique feature of the blue coral snake is its abnormally large venom glands, which produce a novel voltage-gated sodium channel modifier. It is the only known snake to produce a neurotoxic three-finger toxin, specifically one that directly affects voltage-gated sodium channels. Before we take a look at the rather unique venom isolated from the blue coral snake, let's take a moment to define what we mean when we say venom and take a look at some other organisms that make use of this biochemical weapon. Venom can be loosely defined as any toxic substance that is actively injected into prey or a potential threat. This usually occurs through bites or stings. There are a large variety of venoms utilized in the animal kingdom, some more potent than others. However, most of these venoms are used in an effort to either immobilize or kill a target. Some common creatures that have been known to use venom include snakes, spiders, and scorpions. The particular venom we're going to take a look at is named calliotoxin, after Calliophis bivageris.
Calliotoxin is a specific three-finger toxin. These toxins are characterized by 60 to 74 amino acids and 4 or 5 disulfide bridges. When characterizing toxins, the high disulfide bridge to amino acid ratio is a clear indicator of a three-finger toxin, as no other toxins possess such a high ratio. A three-finger toxin consists of 3 beta-stranded loops extending from a central core containing the 4 conserved disulfide bridges. As the name implies, there are 3 major functional sites: the cytolytic site, hypotensive site, and analgesic site. Three-finger toxins are responsible for a myriad of biochemical maladies, including neurotoxicity, cardiotoxicity, and anticoagulation. Here we show 2 graphics showing the structural conformation of a general three-finger toxin. The graphic on the left demonstrates its beta-stranded loop secondary protein structure. The graphic on the right is a nuclear magnetic resonance representation of the relative locations of the amino acid residues present in the toxin. Calliotoxin, also known as delta-elapitoxin CP1A, is a novel three-finger toxin that is a sodium gate modifier and produces spastic paralysis. This form of paralysis is characterized by muscle rigidity and heightened muscle reflexes. This is accomplished by a shift in the voltage dependence of NAV activation, leading to more hyperpolarized potentials, which inhibits inactivation. In contrast, flaccid paralysis is characterized by reduced muscle function due to the absence of tendon reflexes, which can eventually lead to complete paralysis. There is currently no known antivenom to counteract the effects of calliotoxin.
Through the use of Edman degradation and venom gland transcriptome analysis, the primary structure of the venom has been elucidated. As you can see here, there is a heightened presence of cysteines that allow for disulfide bonding. When compared to other known three-finger toxins, calliotoxin presents a distinct amino acid sequence aside from the presence of cysteine in key locations. A mass spectrometry analysis indicates calliotoxin possessing a molecular weight of 6,725.9 Daltons. As I'm sure a lot of you already know, a voltage-gated sodium channel is essential for rapidly transporting sodium ions into a cell. At resting potential, represented by the leftmost figure, these channels are inactivated by an inactivation gate or ball. However, when a nerve impulse travels down an axon, the channel becomes activated and rapid transport of sodium ions is allowed into the cell, as seen in the middle figure. Under normal conditions, the membrane potential would fall back down and eventually end up at resting potential once more. However, this process becomes altered when calliotoxin binds to the voltage-gated sodium channel. Calliotoxin will essentially create a delay of sodium channel inhibition by shifting the voltage dependence of NAV activation. This causes continual influx of sodium ions, leading to severe impairment of muscles.
Researchers examined the effects of calliotoxin on NAV 1.4 receptors in HEK293 cells. NAV 1.4 receptors are a subtype of voltage-gated sodium channels, essential for skeletal muscle function in mammals. Whole cell patch clamp cell recordings were taken upon the interaction between calliotoxin and the NAV 1.4 channels. Their research demonstrated an enhanced current of sodium ions entering the cell and a delayed inactivation of sodium ion flow. This led to a depolarizing shift in voltage-gated sodium channel activity, as well as an increased inward ramp current.
These results demonstrate that calliotoxin acts directly on voltage-gated sodium ion channels. By delaying the sodium channel's inactivation, the sodium channel remains open longer, allowing for a greater influx of sodium ions. This increases the concentration of sodium inside of the cell, allowing for more depolarization and extending the time of the action potential.
Some of the methods these researchers used are techniques that we have studied earlier in the year. These techniques include mass spectrometry and Edman degradation. However, other procedures such as neurotoxicity studies, assay-guided fractionation, and electrophysiology were also utilized in an effort to characterize this unique venom. During the neurotoxicity studies, male chicks were euthanized by carbon dioxide and exsanguination in order to get to their biventor cervices, nerve muscle. These muscles were then treated with calliotoxin and its effects were observed. During assay-guided fractionation, crude calliotoxin was fractionated into 30-second fractions on a column and allowed to flow at a rate of 1 milliliter per minute. This particular technique ended up concluding that calliotoxin was selectively inhibiting NAV channels. During the electrophysiology procedure, the researchers observed ramp currents that were elicited by calliotoxin and demonstrated its ability to alter the voltage-gated dependence of the NAV channels.
After calliotoxin was characterized by the researchers, they identified this blue coral snake as the epitome of toxioferin reptile venom derivation, on account of its extraordinary venom glands and novel venom chemistry. Their study aimed to increase their general knowledge of how venom evolution has proceeded in order to create such a potent biochemical weapon. Their research also shows the importance of using these evolutionary roadmaps as a basis for further study.
While venom is normally detrimental to an individual's health, further research into the use of calliotoxin could prove to produce positive medicinal outcomes. This leads to the final question of whether or not calliotoxin can be utilized as a potential pain reliever. The general idea is that in controlled amounts, this venom may be able to interfere with pain receptors by blocking sodium as well as other ion channels from functioning properly. However, much research still has to go into calliotoxin before effective and safe methods can be established for medicinal purposes. Ultimately, some would argue that calliotoxin can be used to aid in bouts of excessive pain, while others say that this venom would likely do as nature intended and end up further harming the patient. However, one day, venom as a medicine might not seem like such a far-fetched idea to the everyday person. Hopefully, you enjoyed our presentation. We hope you learned enough to stay away from blue coral snakes and how truly important your NAV channels are.